Welcome back, friends. Welcome back again to one more other lecture session from this channel. So in this lecture session, we will be studying regarding the basic differences between Meiosis-1 and Meiosis-2 from cell cycle and cell division in detail. So here the most important objective of Meiosis-1 is nothing but to regulate the pairing between homologous chromosomes and to segregate the homologous chromosomes and to regulate the production of two daughter cells. But whereas the most important objective of Meiosis-2 is nothing but to separate the sister chromatids and to regulate the production of four haploid daughter cells. Is it clear? So let me write it on the board. The most important objective of Meiosis-1 is to regulate the pairing between homologous chromosomes and to regulate the segregation of homologous chromosomes and to segregate homologous chromosomes and to produce two daughter cells But whereas the most important objective of Meiosis-2 is nothing but to regulate the separation of sister chromatids and to regulate the production of four haploid daughter cells. So let me write it here. The most important objective of Meiosis-2 is to regulate the separation of sister chromatids and to regulate the production of four haploid daughter cells and to regulate the production of four haploid daughter cells. Is it clear? And right now we will be studying regarding the basic differences in between each individual phase of Meiosis-1 and Meiosis-2 in detail. So at first, if we consider prophase 1 of the Meiosis-1, right? Prophase 1. So here what exactly happens during the prophase 1? So here during the prophase 1 of the Meiosis-1, pairing in between the two homologous chromosomes takes place. Is it clear? During the prophase 1 of the Meiosis-1, pairing in between the two homologous chromosomes takes place. And after the pairing of two homologous chromosomes, now here genetic exchange or crossing over takes place in between the two non-sister chromatids of homologous chromosomes. Is it clear? After the successful regulation of pairing in between the two homologous chromosomes, now here genetic exchange or crossing over takes place in between the two non-sister chromatids of homologous chromosomes. Is it clear? So let me write it here. During the prophase 1, pairing between two homologous chromosomes takes place. And in addition, genetic exchange takes place in between the two non-sister chromatids of homologous chromosomes. The two non-sister chromatids of homologous chromosomes. Non-sister chromatids of homologous chromosomes. And if you consider the prophase 2 of the meiosis-2, right? Prophase 2. So here during the prophase 2 of the meiosis-2, nuclear envelope breakdown takes place. Is it clear? During the prophase 2 of the meiosis-2, nuclear envelope breakdown takes place, and in addition, spindle fiber formation occurs. During the prophase 2 of meiosis-2, nuclear envelope breakdown occurs, and in addition, spindle fiber formation occurs. So in general, prophase 2 of the meiosis-2 occurs after the successful organization of telophase 1 of the meiosis-1. So here at the end of the telophase 1 of the meiosis-1, in general, two daughter cells are formed. Now here the two daughter cells which are successfully formed during the end of the telophase 1 of the meiosis-1. Now here these two daughter cells in general they participate in case of the prophase 2 of the meiosis-2. Now here in order to regulate the meiosis-2, now here the nuclear envelope breakdown takes place. After the successful breakdown of the nuclear envelope, then after spindle fiber formation occurs. Is it clear? And if you consider the metaphase 1 of the meiosis-1, during the metaphase 1 of the meiosis-1, the specific homologous chromosomes initially which are under paired conformation, they are capable to remain at the center of the cell. Is it clear? The specific homologous chromosomes initially which are under paired conformation, now here these two homologous chromosomes they remain at the center of the cell. So let me write it here. The specific homologous chromosomes initially which are under paired conformation remain at the center of the cell. During metaphase 1. And if you consider the metaphase 2 of the meiosis-2, so here during the metaphase 2 of the meiosis-2 in general, here homologous chromosomes they won't remain at the center of the cell. Instead of remaining of homologous chromosomes at the center of the cell, here sister chromatids, the specific sister chromatids, they remain at the center of the cell. So let me write it here. Metaphase 2. So here during the metaphase 2, during the metaphase 2, the specific sister chromatids of dyad chromosomes remain at the center of the cell. Is it clear? So here this is regarding metaphase 2 of the meiosis 2. Then after we will be studying regarding anaphase 1 of the meiosis 1 in detail. Is it clear? So let me arrange the things here. And if you consider the anaphase 1 of the meiosis 1, so here during the anaphase 1 of the meiosis 1, segregation of homologous chromosomes takes place. That means the specific homologous chromosomes initially during the metaphase 1 of the meiosis 1 in general, they remain at the center of the cell. So here the specific homologous chromosomes initially which are present at the center of the cell, now here these homologous chromosomes, they are ready to segregate or they are ready to separate during the anaphase 1 of the meiosis 1. During the anaphase 1 of the meiosis 1, separation of the sister chromatid does not take place. Only segregation of homologous chromosomes takes place, because during the anaphase 1 of the meiosis 1, splitting of the centromere does not occur. So here, because of the absence of the splitting of centromere, here during the anaphase 1 of the meiosis 1, only the segregation of homologous chromosomes takes place. Is it clear? So let me write it here. Anaphase 1 of meiosis 1, right? So here during the anaphase 1, of meiosis 1, segregation of homologous chromosomes occurs. But whereas during the anaphase 2 of the meiosis 2, so here during the anaphase 2 of the meiosis 2, likewise the anaphase of mitosis, splitting of centromere occurs. So here because of the splitting of the centromere, here the separation of sister chromatid takes place. Is it clear? During the anaphase 1 of the meiosis 1, segregation or separation of homologous chromosomes takes place. But whereas during the anaphase 2 of the meiosis 2, here segregation of homologous chromosomes does not take place. Instead of undergoing the segregation of homologous chromosomes, during the anaphase 2 of the meiosis 2, separation of sister chromatid occurs, because during the anaphase 2 of the meiosis 2, likewise the anaphase of mitosis, splitting of centromere occurs. So here because of the splitting of the centromere, here separation of sister chromatid takes place during the anaphase 2 of the meiosis 2. Is it clear? So let me write it here. Anaphase 2. So during the anaphase 2, of meiosis 2, separation of sister chromatids occurs because of the splitting of centromere. And during the telophase 1 of the meiosis 1, right? During the telophase 1 of the meiosis 1. So here, During the telophase 1 of the meiosis 1, once when the specific homologous chromosomes, once when they undergo segregation, and once when they reach to the spindle poles, now here nuclear envelope formation occurs. So here after the successful formation of nuclear envelope, here at the end of the telophase 1, here we can observe two daughter cells. Is it clear? At the end of meiosis 1, the two homologous chromosomes, once when they reach the spindle poles, nuclear envelope formation occurs. Which results in the formation of two daughter cells. Right? Now these two daughter cells we successfully formed at the end of the telophase 1 of the meiosis 1. Now here they are ready to enter into the prophase 2 of the meiosis 2. Is it clear? And if we consider the telophase 2 of the meiosis 2. So here what exactly happens during the telophase 2 of the meiosis 2? So as I have already mentioned you, during the anaphase 2 of the meiosis 2, splitting of centromere occurs. So here when the splitting of the centromere occurs, now here separation of sister chromatid takes place. So here after the successful separation of the sister chromatids, to these sister chromatids, nuclear envelope formation occurs during the telophase 2. Is it clear? So let me show it here. So let us we assume it has one specific chromosome, one dyad chromosome, right? And it has the other dyad chromosome. So here when the splitting of the centromere occurs, during the anaphase 2 of the meiosis 2, right? When the splitting of the centromere occurs during the anaphase 2 of the meiosis 2, now here separation of sister chromatid occurs. Is it clear? So let me show it here. So these are nothing but what? The sister chromatids. Separation of sister chromatid takes place. So here to these four sister chromatids, nuclear envelope formation occurs or nuclear envelope formation takes place during the telophase 2 of the meiosis 2. So here at the end of the meiosis 2, here we can observe four haploid daughter cells. Is it clear? So let us assume it as the nuclear envelope. So here all these are nothing but what? The four haploid daughter cells. So let me write it here. During the telophase 2 of meiosis 2, the specific sister chromatids which are formed during the splitting of centromere, to these sister chromatids, nuclear envelope formation takes place. which results in the formation of, which results in the production of 4 haploid daughter cells, which results in the production of 4 haploid daughter cells, 4 haploid daughter cells. Is it clear? So here this is regarding the basic differences in between Meiosis-1 and Meiosis-2 in detail. And in the next upcoming lecture session, we will be studying regarding numerical based questions from cell cycle and cell division in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.